Hey everybody, it's Miss Clemmy. Sorry I can't be there today. I know you're probably looking forward to me being there, but I can't. So we're going to carry on as usual though. And so in your folders, you should have a half sheet that looks exactly like this table in blue. I need you to get that out and simply fill it out with using your notes from last class. And you should have, basically you're writing definitions for each term and putting a little check mark by which groups of organisms that adjective fits. And so I'm going to have the sub simply start a timer and hold you to that. So about 7 minutes. And you can pause the video, and when you're all done, let's come back. All right. Good luck, and go. All right. Welcome back. So hopefully this should have been a pretty easy review for you. So let's take a look. The first term, eukaryotic cells. Complex cells filled with lots of organelles. Now the opposite, prokaryote, would be the simple-celled organisms. And so if we can, let's do some check marks here. Eukaryotic, of course, animals and plants, fungi. Protists are all eukaryotic. And that leaves just bacteria as prokaryotic. And viruses don't belong in either because they're not native cells. Moving on, heterotroph, organisms that must consume food for energy, like us. And the opposite, autotroph, organisms that make their own food for energy. So we can check off us in the animal kingdom. Fungi, which makes them very similar to animals. Protists must consume food, and so must bacteria. So obviously the opposites, plants, we know they photosynthesize, so they're autotrophs. But surprisingly, some protists and some bacteria, depending on the species, can also photosynthesize. And the last two sets of terms, unicellular versus multi. One cell versus many celled organisms. Find my unicellular. We have protists can be unicellular as well as bacteria. For many cells, obviously animals like us are, plants are, fungi are, and here's the oddball again, some protists can be multicellular as well. Notice viruses don't belong in any of these categories because they simply are not made of cells. So I hope you did well on that particular half sheet. Put that back in your folder and keep that for a later date. And so let's move on. All right, at this time, we're going to transition a little bit here. I'm going to have you pick up the pathogen introduction, looks like this, and a little disease card that should have a different disease for everybody in the room, or pretty close to it. And at this point, then you can go ahead and pause the video. Just make sure that you're ready to play the other video on the computer screen from that time you see here, starting at 2 minutes and 16 seconds. I'll be back. And I'm back. So that was a little introduction on pathogens, on things that cause disease. And we're going to focus on influenza or other viruses that cause disease. So at this point, we're actually going to use the pathogen intro sheet. And you guys are going to fill in, first of all, you're going to read the introduction, and I'll have the sub read that aloud. And make sure we understand those terms in bold. Then you're going to fill out the second column for your disease card. So what is the cause of your disease? How does it get transmitted or spread? Is it re-emerging or is it endemic? So fill in that second column. Then when everyone's done with that, you can turn back in your disease cards, and then you have to fill in the rest of the table. So you can find a different person with a different disease, and make sure you fill up the table, and we can look at those patterns and trends. Again, we're going to hold you to a timer. So let's say about 2 minutes to fill in your disease card and give that disease card back to the sub. Then we'll give you about 6 minutes to fill in the rest of the table with your friends. Pause the video. Good luck. And we're back. So I hope you kind of found that interesting to see all these different types of diseases. They could be caused by fungi. They could be caused by bacteria or viruses. And so that kind of leads us into the big picture, which is our unit we're starting. And we've kind of had some dabbled in all these different organisms, but we're going to really focus in on one group today. Unfortunately, if we look at the entire tree of life here, it's not even on here. And so, we could say, well, these are the prokaryotes and everything else is eukaryotes. You can see, some plants, some fungi, insects, starfish, fish, mammals, birds, all of those. We're going to study all of them. But we want to start at the very beginning, things that aren't even on this list. And so we're going to start by looking at viruses today. So here we go. All right, at this point, if you need to pause, I'll need to have the next sheet handed out, which is simply called Viral Structure and Function. So if you need to pause, especially to write down the learning target, please do so. And I'm back. So you should have jotted down the learning targets. We're going to look at the impact of viruses. Now, it's going to all lead up to this super awesome excerpt we're going to read, highlighted here, called The Hot Zone. That's going to be next class, but we got to learn a little bit about viruses first. So we're going to take a look today to learn the different structures that make up a virus and what those structures do. Okay, so that's going to be our focus today. Here we go. All right, so before we read The Hot Zone, we're going to answer 3 questions. What exactly are viruses? How do they make more viruses? And how do we treat them and prevent them? Here we go. Question 1. So you guys have a short paragraph on your notes. I'd like you to underline all of the structures in a virus. And then you're going to go ahead and transfer those underlined structures into Table 1. I'd like you to do this on your own independently. So please pause the video, but let's watch the clock and let's take 5 minutes. So pause the video now. And we're back. All right, so you guys should have filled in Table 1. I'm going to go over Table 1 and the answers, but at the same time, if you look down at Table 2, I'd like you to add labels to it. So we're going to begin with the first thing that's made of a virus, and it's genetic material. You guys know what this is. It's DNA, and in some cases, like HIV, that virus uses RNA. Okay, it just stores information for that virus. And so you're going to go ahead and label a bunch of those squiggles there as the genetic material in your Table 2 box. Then we're going to add a layer on. We're going to add a protein code. Please label this on your Table 2 and check. Now here's where I'm having you multitask. Check the function of the protein code in Table 1. Make sure you got the right information down. And finally, the third component of a virus, and my favorite, are the surface proteins. These have the virus latch onto a host cell. It could be one of our cells. It could be another organism that it's infecting. Okay? Label these on Table 2 and also check your function on Table 1. When you're ready, you can go ahead and flip over that sheet. Because that brings us to question 2. How do viruses reproduce? Now, humans, excuse me, they reproduce by using meiosis and fertilization and mitosis, but viruses aren't us. They have their own way of doing things. They have 2 different cycles, the lysogenic, which is going to be the second one we look at, and the lytic cycle. Okay, so we're going to go ahead and look at the first one up there, the lytic cycle. Please read over the couple sentences and take a look at the picture. I want you to discuss that picture. Can you actually make sense of that cycle? What's going on with that little sunburst that is a virus? What is it doing to the host cell? So pause and discuss. Take 90 seconds. Go. And we're back. Here's the lytic cycle. So if I write something, you write something. Here we go. So here's our little virus. So let's label this guy. And this is our host cell. So we got to latch on with the surface proteins, but then the little virus injects its DNA. So viral DNA. And it uses that host cell like a little virus factory to pump out and make, I'm actually going to label it, it's like a virus factory. It's making tons more viruses, and before you know it, The host cell can't hold those viruses and it literally bursts open, spills out those new viruses, and so the cycle starts over. This one can go to a new cell, new cell, new cell, and spread the virus all over. So pause the video again and see if you can't summarize that particular process, the lytic cycle. I'm going to put up my summary, but by no means should yours be nearly as long as this. Pause. Good luck. And we're back. We're going to move on to the last cycle, the lysogenic cycle. Again, read over those sentences and see if you can't figure out what's going on. You're going to see some similarities to the lytic cycle. In fact, after about 90 seconds and you've shared and examined the picture with your neighbor, see if you guys can have a discussion on how this cycle looks a lot like the lytic, but there's a few different subtleties. Good luck. Pause the video. And we're back. I hope you guys could figure out some of those steps. So here we go. Again, if I label stuff, you label stuff. So here we go. We have the virus. And we have our host cell, but something else is in here this time. This is the host DNA that's going to play a role. And so the same couple steps begin. We have the virus latching on, injecting its DNA, but here's where it begins to diverge. Now we have the host cell. The DNA and the viral DNA kind of mixes together. It says, you know what? I'm going to just slide in here and kind of take a back seat for a little while. No one's going to know I'm here. And so when the virus or the host cell replicates through mitosis to make two cells, those two cells have a copy of the viral DNA. And those two cells are going to make more copies and replicate the viral DNA, and so on and so on. And so the virus is kind of quietly taking over. And then at a moment's notice, the virus DNA comes out of the host DNA and starts making copy after copy after copy. And eventually, the cell can't hold them, and we start the cycle over. So this cycle takes a while, and that virus kind of remains inactive for a little bit, but essentially, it kind of looks like if you go, well, about here, this is kind of the lytic cycle, so it shares some properties. It kind of takes a detour, if you will. It takes a longer route to the lytic cycle. That's the lysogenic cycle. All right, and finally, one last discussion with your partner. How are these viral infections like the flu treated? Those are your guiding questions below, and see if you can have a good discussion as a whole group. So pause the video at this time and discuss. And we're back. Now, how are they treated? Well, unfortunately, when you get the flu or the cold, we can't treat the virus. We can't kill it. Our immune system are the only thing you can kill it. I can't give you medicine for that. When you take medicine for your aches and pains and your stuffy nose, you're actually just treating the symptoms caused by the virus, not the actual virus. Now, on the other hand, if you want to be proactive, if there's a vaccine, you can prevent viral infections. You just can't treat them. But we'll talk a lot more about this at a later date. Okay? Now, at this point, we're just about finished. Now, go back to that first sheet, the pathogen sheet. On the bottom, it says step 3. I'd like you to answer all those questions, and you're simply just going to put A, B, or C for all of those. But before you can leave this room, I want those answers perfect. Do them by yourself. Use your notes, but then go and check your answers with self. Thanks for listening, and good luck.